Yeah, it's a pleasure to be back. It's been a long time since I was here, it's both familiar and unfamiliar all at the same time. So I've been really enjoying my conversations with everyone. And how do I work this thing? Just hit that? Okay. So what I thought I'd do is step back a little bit and try and set the scene for some of the things that we've been thinking about and why we've been thinking about them. And in the light of the conversations I've been having, maybe that's actually kind of a good idea. So I thought I might tell you how we've been thinking about what theory in biology means in an age where we have the onset of AI and machine learning. So I think we're all familiar with the fact that there are two trends that are converging sort of explosively in modern biology. One is that we're drowning in data, and biologists have been very successful at developing increasingly sophisticated methods to extricate data from living organisms. And that process is not slowing down, it's going faster. At the same time that this is happening, and I'm just repeating something which everyone knows here, so I won't elaborate it. At the same time, something else which we're all familiar with is happening, which is that computer science has gone through a kind of quantum leap in terms of artificial intelligence, and that's perfectly evident. For instance, in last year's Nobel Prizes, which in chemistry went, not surprisingly, to Google DeepMind for the development of AlphaFold. I don't think that came as a surprise to anyone, but clearly I would have loved to have been present at the conversations in the Nobel Committee about this, but the physicists didn't want to be left out. The awards in physics also went to two of the pioneers in developing some of the infrastructure for what DeepMind eventually did, particularly in the context of neural networks. So keep your eye on John Hopfield, because he'll reappear later in my story in a different guise. So these two trends are colliding with, I think, enormous impact in biology, which I think is visible in many of the things that you read, both in the popular press and in the literature. And I've just extricated two of the more extreme statements from things that perhaps you're familiar with. The end of theory. There's no need to do it anymore because the machines have made it obsolete. And we see the founder of Facebook and his wife hoping to eradicate human disease by 2000. So it's very ambitious statements that are being made, and one begins to wonder what the role of theory is in biology in the context of these claims. Now, as Tom mentioned, in a previous life, I used to work in the computer world, and I know some of the people who are involved in this kind of very interesting transition that's taking place. And when I want to annoy them, I kind of compare them to the aliens in the film Independence Day. Now, I'm sorry, I have a very lowbrow taste in films. So if you're not familiar with this, this is a situation in which a really horrible alien race goes from planet to planet, think of scientific domain to scientific domain, and kind of consumes all of the life from it. And once consumed, it sort of moves on to the next phase. And computer science is in the process of sort of doing this with biology. Now, in the fictional world, the unlikely combination of Jeff Goldblum and Will Smith saved the world for humanity. I don't think it's going to be quite so simple in real life. But joking aside, what is there to be afraid of in terms of what is happening now? And I think there are two parts to it. And one part is something which one of the pioneers in reinforcement learning theory, Rich Sutton, wrote about, and which he describes, I think, very aptly as the bitter lesson. And what is the bitter lesson? Well, the bitter lesson is that if you want to make a machine that, say, looks at an image and sees a cat, or hears acoustic recording and deciphers the human speech in it, what people used to do was to try to mimic how humans did that, how the human visual processing works and human auditory system works. And that kind of works, but not really. And the bitter lesson is the recognition that actually that's the wrong thing to do. What you should forget is anything to do with the actual mechanism of how biological systems work. All you need is a vast amount of data and a general purpose training algorithm. And that's precisely what large language models are. Now, there's a consequence of this. It works extremely well. It simulates the understanding of language, but it's a simulation. And I've just extricated here from a recent blog post a very interesting conversation, not a conversation actually, a response that ChatGPT gave to the question, a man and a goat are on one side of the river, they have a boat, how can they both go across? Now you're familiar with these kinds of trick questions. This is not really a trick question, it's kind of very straightforward. And ChatGPT answers, to get both the man and the goat across, they follow these steps. Take the goat across the river first and leave it on the other side. Now, that's actually, clearly the machine has not understood the question. And of course, it gets more hilarious as you read on. And the point here is something that should be obvious, but kind of perhaps gets lost in conversation, is that large language models don't understand language. And this is not a bug. This is a feature. Okay, so the point here is that what is happening in AI is kind of predicated on forgetting about mechanism. Now, there are all sorts of things that you can say, ifs and buts, to answer that statement, but that I think is one of the driving things behind this, and this is the bitter lesson, with the emphasis on bitter. Okay, so the other thing, and this is now much more specific to biology, is the fact that data are contingent. They depend on the technological resources that we have at any particular point in time, and they represent, if you like, a kind of narrow biased projection of the kind of underlying reality that's really there. And if you if you look at the biological literature over a historical period, what you will see is that the kind of stories that people tell about what the data is saying change as the data, the way in which we acquire data changes. And this is just one example. There was a point in time in which if you wanted to look at proteins in cells, what you had to do was to grind up 1 million cells and use a Western blot. And if you do that, you tend to make the assumption that what you see, which is an average, is representative of each of the individual cells in the population. And what is true is that sometimes that's definitely not the case. That instead of having a situation, as you see in part B here, this is taken from a well-known paper from Jim Farrell, where essentially every cell is doing the same thing, which is around the average. What is actually happening is that every cell is doing something different. It's either doing yes or no. And what you see in the average is completely unrepresentative of what you see at the population level. Now, when I tell you this, this is kind of obvious. Everyone knows about noise and heterogeneity. But if you replay the tape of what was being said when there was no single cell measurements to be made, some of the stories that were developed on this basis were essentially delusional. Now, this is actually the normal practice of biology. And because biology tends to have a very short-term memory, this is not how biologists think of their own discipline. But in fact, if you go back through the historical literature, what you will see is that the stories change depending on the kind of data that's available. So now you have, this is normal biological science colliding with the developments of AI. So you have, I think, a recipe for telling increasingly sophisticated delusions. Okay, and so from where I sit, this is one of the challenges that we face in the way the world is at the moment. And so the question for me as a self-confessed, well, a mathematician, I still identify as a mathematician, theorist in biology is, what do we do? And so, and I think one answer is actually to go back to physics and look at what happened in physics. And this is a very interesting quote from Einstein. Some of you may know. It's actually not directly a quote of his. It's attributed to Einstein by Werner Heisenberg in one of his books. What Einstein is saying here is that it is really theory that tells us what we can observe. And so this dialogue with Heisenberg occurred in the context of a conversation about the foundations of quantum mechanics. And it's probably well known that Einstein was very opposed to the conventional sort of Copenhagen interpretation of quantum mechanics. But leaving that aside, I think perhaps a better way to think about this in the context of Einstein's work on Brownian motion. Because when Brownian motion was discovered, which is in the late 18th century, it was really a mysterious phenomenon. You put little pollen particles into fluid, and suddenly these particles are moving around. Where's the motion coming from? And over the course of the 19th century, vast amounts of data were acquired about Brownian motion in different contexts. Many theories were developed. It was complete confusion until Einstein developed the understanding that Brownian motion is a consequence of atomic fluctuations. And that was really the basis for showing that atoms existed. So this is, I think, a very good illustration of what Einstein meant when he said that it is the theory that tells us what we can observe. But the moment that theory became real, all of Brownian motion became explainable. And in its absence, it was mysterious and led to all sorts of delusions about what was causing that underlying motion. So physics went through this kind of transition probably in the early part of the 20th century when Heisenberg and Einstein were arguing about this. In physics, it is theory that determines what you observe. It is theory that says you have to build a huge collider, costing the GDP of a small country in order to observe the particles that the theory predicts. Biology is still in the realm of developing its conceptual language from data. I think perhaps I would argue that maybe it is going through the transition in neuroscience, arguably maybe in immunology. But I think the question for us as theorists is, is biology or molecular biology, cell biology always going to be driven by data? Or is it in some time, at some point, going to go through this transition of being driven by theory? And so I think the question is, can we get behind the data? Can theory help us to see what the reality is behind the particular projections of the reality that we acquire at the moment? And I think the answer, is yes. And in the last few years, I think some of the things we've been trying to do in the lab has been with this in mind. So this is really a sort of very long-winded kind of trying to explanation for the approach that we've been trying to take. And I think this attempt to try and get behind the data has taken sort of two directions for us. Sorry, I keep going backwards here. One direction, which I'm going to talk about today, is around energy and the way in which it plays a role in the biological context. And the other is information. And I will just use a slide to say that actually this other direction of information is, I think, very interesting in its own right and is playing out for us in terms of trying to think about learning, the sort of cognitive science idea of learning in the context of individual cells. We've been doing a lot of work on this. We have an ERC synergy grant with 3 of my colleagues, some of whom you may know, to specifically work on this. So I'm very excited about this, but it is not entirely detached from what I am going to talk about today, but it's kind of in the background, okay? But today I want to say something about the other direction that we've been taking to try and get behind the data, which is to do with energy. Okay, so we know a lot about energy. Clearly, life is a non-equilibrium phenomenon. And we particularly know a lot about energy in the context of situations where the energy does mechanical work, causes patterns to form, generates force. Where we know somewhat less about the role that it plays is in the context of information processing. And just to give you a sort of, kind of little hint about that, maybe one of the reasons for that is actually you can process information without using energy. In fact, you can Turing compute without using energy. You have reversible computers. So information processing is more subtle. What role does energy play in that context? And in particular areas of biology, and one of the areas we work in is gene regulation, a lot of our thinking, many of the thinking of experimental biologists, is still grounded in equilibrium assumptions. And I think that's actually something that's going to have to change. So what role does energy play? And one of the people who really, I think, gave us very important insights into this is the person who's just won the Nobel Prize in physics for something very different, and that's John Hopfield. And this goes back to a very famous paper of his on kinetic proofreading, which, if you're a biologist, every molecular biologist knows about kinetic proofreading. And what was Hopfield trying to say here? Well, he was thinking about the problems of error correction, of correcting errors when you're synthesizing polymers like DNA or RNA or protein. The kind of thought experiment he did was to think about how do you discriminate between a correct substrate to incorporate in your polymer and an incorrect one? And the first way you do it is actually by binding. Well, and this doesn't require any energy, but typically if you have a correct substrate, it tends to stick for longer. It has a slower off rate, and that's typically the case in biology, that binding discrimination takes place through the off rates. And so what you have is a situation in which binding allows you to get a certain amount of error correction. You have a kind of, the probability of binding an incorrect substrate versus the probability of binding the correct substrate has some small value, epsilon. And if you do the calculations for most of the things that you encounter in biology, it's about 1 in 10 to the 3, 10 to the 4, something like that is the kind of error rate that you would expect. In fact, measurements show that replication and translation and transcription have error rates of 1 in 10 to the 10, 10 to the 9. How do you get such a huge increase in the error rate, the accuracy? And Hopfield said, well, if you can get an error ratio of epsilon by doing 1 binding, how about doing 2 bindings? So you just move to another state, which allows you to do another discrimination. If you have an error ratio of epsilon with 1 binding and an error ratio of epsilon with another binding, shouldn't that give you an error ratio of epsilon squared? And what's the problem with that argument? Well, the problem is that if the system that does the 2 bindings is operating without energy expenditure at thermodynamic equilibrium, it cannot tell whether it's gone through 1 binding or 2. And I shouldn't probably have to do this, but just to remind you, I'm a mathematician, it took me a long time to understand this stuff. We tend to think about things called steady states, where nothing changes over time, and physicists know well that there are 2 kinds of steady state. There is the steady state on the left, which is the state of thermodynamic equilibrium, and magical things happen at thermodynamic equilibrium. It's extremely special. If you take a movie of the molecular level, you can't tell which direction it's going in. And in particular, there is no path dependence. So in the picture that we had on the other side, it's impossible to tell whether you took 1 bite of the cherry or 2 bites of the cherry. The only way you can do that is if you actually expend energy, take the system away from thermodynamic equilibrium, you have a flux through the system, it's still a steady state, but now you have path dependence. And now you can tell whether you took 1 bite or 2, and then you can get an improvement in the error ratio, which is not quite epsilon squared unless you're in some limit, but it's close. So this is Hopfield's insight. I think it's a very important insight, and certainly it had a huge impact in biology because kinetic proofreading is thought to play a role in all sorts of contexts of biological discrimination. Okay, but I think there's actually more that Hopfield had to say to us here, which I would extract in the following way. I think what he was really saying is that if you have any information processing task, which is not copying a polymer, but anything that you might imagine, then if the system that is implementing this is operating without energy expenditure, it's in a state of thermodynamic equilibrium, then there's a hard upper bound to how well it can do that task, however you want to measure how well it does it. And that's set by basic physics. It's basically detail balance. And there's only one way that you can get beyond that. You have to inject energy, you have to take the system away from thermodynamic equilibrium. And then, if you read Hopfield, there's kind of almost a kind of folk assumption that if you throw enough energy to it, you can improve it as much as you want. And I don't think we actually know that that's true, or there's any theorem to back that up. But anyway, certainly, if you want to improve it, if you want to get past that bound, there's no way you can do it without expending energy. And I think we call this limit of what you can achieve at thermodynamic equilibrium a Hopfield barrier. And I think for us, this has been a way of trying to think about information processing, again, as a way of getting behind the actual data to what's really going on under the hood. And so the problem of characterizing the Hopfield barriers for various information processing tasks is something that we think is actually a strategy for sort of thinking about biology differently. So that's one of the things that we've been trying to do in the lab. Okay, so what I want to talk you through is some of the consequences that we found of thinking in this kind of way. And I'm going to come back to the Hopfield barrier in a moment. But what I want to specifically talk about are input-output responses. So what do we mean by that? So I'm going to work through several examples here, but I just wanted to set the sort of mathematical scene for how we should think about this. So imagine we have some kind of biological system. It's confronted by many kinds of inputs, many kinds of influences from outside. Some of these influences we think of as inputs, and by that I mean only that as an experimentalist, we have control over, say, the concentrations that are involved in those things that are impinging on the system. And there are also other things that might be interacting with the system which we are not actually controlling. So we might think of those as sort of co-regulators. So this is a very generic kind of setting. And then the system itself, we imagine, and this is where, these are the actual sort of underlying mathematical assumptions, that it's Markovian, it has no history. And the way in which we're interacting with it is that we're changing it quasi-statically, right? So we allow the system to come to steady state, we measure some output, and then if we want to make a change to the input, we do it in small steps, allowing the system always to come back to its steady state. So that's what it means to be quasi-static. And the output is some kind of linear combination of steady state concentrations or probabilities that are normalized in this way. Okay, so this is just the sort of technical details of how we're going to think about these input-output responses. So that's all very abstract, but that's the actual underlying mathematical setting.
Are you assuming that it's steady state? Are you assuming it is or isn't an equilibrium steady state?
Just a steady state. It could be equilibrium, it could be away from equilibrium. And there both examples exist. Okay. All right, so you all know examples of this. This is the first one that any biologist encounters. It's the Michaelis-Menten rate formula for an enzyme converting a substrate to a product. And just to remind you of the history here, the first sort of studies about enzyme behavior, enzymes were able to do this conversion of substrate to product. And when people first began to acquire data on it, they were surprised to find that when you look at the speed of the reaction initially, before there is product formed, then what you see is this hyperbolic dependence on the substrate concentration. And the reason for this was a little mysterious to begin with until Michaelis and Menten put forward the hypothesis that what happens is that the enzyme binds to the substrate to form an enzyme-substrate complex, which then decays to form the product. And then if you do some very nice mathematics, which is in their original paper, what you can deduce is a formula for how the product rate of product formation depends on the substrate, which you've all seen, I'm sure. This is the Michaelis-Menten formula. And it's based on the assumption, which is very natural in the in vitro biochemistry context that we're studying, that basically the enzyme substrate complex forms very rapidly and then is in a sort of steady state. Okay, and the algebra that you need to go from one to the other still strikes terror into the heart of undergraduate biochemists. Okay, so what we see here is two kind of interesting phenomena. One is that there are 4 quantities on the right, and there are only 2 quantities, sorry, on the left, and there are only 2 quantities on the right. Some of them have disappeared, they've been eliminated. And what we see on the right is as a rational expression in the substrate concentrations. It's a ratio of 2 polynomials. These are first degree. And I'll just point this out. I won't belabor this point, but actually, if you dig into what the Michaelis-Menten constant looks like, the Km, then you might recall, if you've it's not lost to memory here, that too is a rational function of the underlying rate constants. So this doesn't seem like a very interesting thing to remark on. It's just a function, so why can't it be a rational function? But this is the first illustration of how these kinds of input-output responses have this rational form. So I will make a quick aside here that in the context of what we were talking about getting behind the data, is that the enzyme substrate complex was a hypothesis that Michaelis and Menten put forward. And it was so successful that biochemists promptly took it for granted that enzyme substrate complexes existed. And they used it, and the whole use of enzyme substrate complexes just, kind of exploded in the literature, became much more complicated and sophisticated. And it was 30 years before the enzyme substrate complex was actually confirmed to be an actual chemical entity, and its binding and unbinding rates were measured by Britton Chance, who developed the stop flow methods that you need to actually isolate an enzyme substrate complex. So you should kind of compare this with physics, right? Because in physics, if someone comes up with a new particle on the basis of theory, it's not that everyone sort of says, oh yeah, great idea, we'll use this and do physics on the basis of it. You wait until the experiment is done that confirms that the particle exists, mostly. I'm sure there are... Yeah, it's not the official way of thinking about it. But what I find is very interesting. I mean, Chance clearly knew that he was actually providing the first confirmation for the theory that had been put forward 30 years before. And so if you actually read, and this is not an isolated example, I think it's actually extremely commonplace in biology. And so from this point of view, I think it's fair to say that biology is more theoretical than physics. And if you want to sort of follow that, there's an article here that you can have. But this, I think, is a good illustration of how good theory, Michaelis and Menten, allows you to get behind the data, to see what's there that's not actually present in the data, okay, which is confirmed much later. It's very similar in that respect to Einstein's work on Brown induction. Okay, so that was the starting point for a lot of work on enzyme kinetics. And if you read there are thick tomes on more and more complicated enzymes, which have more and more vast numbers of enzyme substrate complexes. And in all cases, what you get is the similar kind of results. So there are systematic ways to extricate from these kinds of reaction diagrams the rate of dependence of product on now multiple substrates. And what you see is, again, the emergence of these rational expressions and the elimination of a lot of the enzyme substrate complexes. So that's biochemistry, but then we come to another very interesting episode in biological history, which is the development of allosteric, the notion that the behavior of proteins is governed by an ensemble of conformations to which things interact with different affinities. This is very famous. This is the work of Monoweim and Changeau and others. And here too, what you have is a situation where you have an ensemble with ligands that interact with it in different conformations, and you can go through some very elementary algebra and discover that the output that you're looking at, which is something like, say, the saturation of binding, is a rational function of the concentration of the inputs, the ligands that bind. Okay, so yet another example. And if you're familiar with the area of pharmacology, this kind of mathematical analysis has been used in pharmacology ever since the very early days in the early part of the 20th century, and is very sophisticated now. It's used for drug design. And these are two examples, one from the context of studying G-protein coupled receptors. This is part of the work that led to Bob Lefkowitz's Nobel Prize. And in his lab, they were habitually using these kinds of models to understand how G-protein coupled receptors were working. And as you can see, again, you end up with the same kind of phenomenon. This is a much more complicated models that are used in the context of pharmacology. And you see the same process of deriving these kinds of rational expressions. Here's a slightly different context. This is ion channels, for which you can do very exquisite single molecule measurements using patch clamps. And now you're looking at the current through a single molecule. And here too, you're understanding how that current depends on the interaction of the ion channel with all sorts of substrates and agonists and inhibitors and all the rest of it. And similarly, you get these same kinds of things now in a single molecule stochastic context, rational expressions of this kind. Another area which you might be familiar with is gene regulation. In the bacterial setting, where the first kind of studies of gene regulation were done, you find these equilibrium statistical mechanics being used to construct models of expression as it depends on the concentration of transcription factors. And the formula that you see on the top right there doesn't look immediately like a rational function. That's because physicists typically use Boltzmann formulations to write down their models, but actually, if you look behind it, it's clearly a rational function of the concentrations of the transcription factors. And this kind of thermodynamic formalism has been developed a lot, particularly by Rob Phillips and others, and is widely used not just for the bacterial setting where it emerged, but also in the eukaryotic setting, for instance, in Drosophila. And in this case, you're relying on equilibrium assumptions. These are situations where you assume there's no energy expenditure, but there are also situations where energy is actually being used. This is a study from Erin O'Shea's lab where one is interested in the interaction between the transcription factor and nucleosomes, and the nucleosomes are being moved around by motor proteins. It is an energy-driven system, and here you get a very complicated setup, which I haven't actually shown you here, so complicated that in order to solve the equations, they had to throw it at MATLAB. So nobody had actually written down what the actual expression was. But if you go and poke around and look at what it is, it's another rational function, which you get by a similar process. So I've gone through this just to illustrate the widely different areas in which these kinds of input-output functions have been obtained. If you read this literature, you will find they're obtained by a whole set of very different methods, very ad hoc. There's a little bit of systematic process using equilibrium statistical mechanics. The enzyme kinetics people have a way of doing it systematically, but they don't understand how broadly it applies. There's a lot of ad hoc methods that depend on the symmetry of particular models and so forth. So you wouldn't imagine that there was a kind of underlying unity behind all of this. And so one of the first things that I want to say is that, in fact, all of these things essentially emerge from one process. And in order to understand that, it comes to see that you have to use a framework that we've developed in my lab, which we call the linear framework. There are review papers on this. I'm actually not going to get into the details of this. It's based on a graph-theoretic approach. So for us, graphs are the kind of fundamental mathematical context that can be used to study all of these different settings. But I'll just tell you what the consequences are of using this. And so the first is what I was leading up to, which should be sort of fairly obvious, is that all of these input-output functions that I've told you about can all be derived in one way, starting from the appropriate graph. And what you see if you run this process is that these input-output functions are invariably rational functions of their inputs. And the coefficients in the rational function are themselves rational functions of the underlying rate constants in the Markov process. Okay, so despite this kind of heterogeneity and diversity and different kinds of complexities and different experimental methods and different data that have been acquired in all of these different settings, there's a surprising unity under the hood. Okay, essentially, it's all the same. Now, this is not how people understand it yet. This is certainly not in the textbooks, but that's really what is the case. So, somewhat to my surprise, as much as anything else, that there is an underlying mathematical unity here, despite the enormous heterogeneity and diversity that we typically see in biology. And that's not typically the story that we usually encounter. Okay, so this is the first kind of universality result that I wanted to sort of mention. Okay, so how are we doing for time?
About 15 minutes.
Okay. Okay, so where are Hopfield barriers gone? I started off by telling you about thermodynamic equilibrium, and I haven't even mentioned it here, right? These systems might be at equilibrium, they might not be. They're all rational. And I've not told you something, I've not talked about a particular rational function that, again, all of you will know about. And it's the Hill function. Okay, and this was a class of rational functions that Archibald Vivian Hill, one of the pioneers of biophysics, kind of dreamed up. He basically guessed that it would be a very good way of fitting data on oxygen binding to hemoglobin. And he sketched a little kind of argument for why it's not, you know, it's reasonable to look at this. It's not a believable argument, although people repeat it. So this is really a kind of, just a kind of guess. It's a family of functions. It turns out, as we know for 100 years, that it's very good at fitting experimental data. It's surprisingly good at fitting experimental data. And we also use it habitually in modeling, where we like having a kind of sharp sigmoidal response in order to generate sort of limit cycle oscillations or bistability or whatever. It's habitually used in theory. But where do the Hill functions come in this story? Because the first thing you realize is that you can't generate them as a bona fide cellular input-output function in the setting that I described. You can approximate them, but you can't generate them on the nose. And that's where the Hopfield barrier comes in. So what you find is that if you now restrict attention to systems where they're operating in a steady state of thermodynamic equilibrium, then it turns out that you can't generate all rational functions that way. That there's actually a very significant restriction on the kind of rational function that you get. The restriction is in the degree of the denominator polynomial. And what it says is that if you have n inputs and input ligands, and they bind at B1, B2, up to Bn sites, just the number of sites of binding that they have, then if your system is working at equilibrium, any cellular input-output function you generate, the denominator polynomial is going to have degree less than or equal to the sum of the numbers of sites. It doesn't matter how complicated it is. That limitation is effectively the Hopfield barrier, as we'll see. Okay, so what does this mean? I mean, this is a very abstract mathematical statement, and what it implies is a limitation on the sharpness of the input-output functions. And the way in which this emerges requires a little bit of unwrapping. And this is where the Hill functions come in. So how do you measure the sharpness or steepness of an input-output function? And that's actually kind of a complicated story. The way we're going to do it is kind of as the most limited way that you can in order to get something interesting. We're going to have two measures of how sharp a function is. And the measures are what we call position and steepness. And I wish we hadn't used that terminology, but now it's stuck. Okay, what are the position and steepness of an input-output function? So very quickly, If you have an input-output function, you normalize it. You normalize the, because it's concentration, you want to get rid of the units. And the way we normalize it is we normalize it to the concentration value which is halfway between its maximum and its minimum. Okay, and that's what the mathematics in the middle of that slide is basically saying, if you can unwrap it for yourselves. And then what we do is to measure for the normalized function the position where the slope is maximal. The absolute value of the slope is maximal. And that's the steepness. And the normalized value where that occurs is the position. Okay, so these functions can be very complicated. They can have many ups and downs. So this is a very kind of limited way of thinking about the sharpness, but it has two measures. It's intrinsic to the function, and there are two of them. It's very important that there are two. Okay, so if we have position and steepness, and I look at rational functions which have that limitation in their degree of the denominator polynomial, I measure position and steepness, it turns out that they can't occupy the whole of the positive quadrant, that they're limited to that gray area. And the gray area is what we would call effectively bounded. They have these wings that go off, they asymptote on the axes. But what happens in the middle is what's really interesting, because this region has this strange cusp. And guess where the cusp lies? It lies exactly on the position steepness locus for the hill functions. And this is for a function of one input variable. If it has 4 binding sites, the tip of the cusp is just below the point at which the hill function has coefficient 4. And if you push the numerics, it gets closer and closer to 4. So what does that mean? It means that the hill function is the barrier to where you are at thermodynamic equilibrium. If you're at equilibrium, you can't get past that, irrespective of how complicated your underlying model is. So that's actually kind of powerful, because let's suppose you get data on position and steepness, which you can get in Drosophila. I'm showing in the insert here some data from my department colleague Angela De Pace, who's a Drosophila geneticist. And what you see, that's actually the little dots there are actually from individual Drosophila embryo for a particular gene. This is the hunchback gene. And it's a synthetic system, which we think is well represented by the model that in this case, 4 sites. And what you see is the data are outside the region. Right, well outside the region. So this is part of the accumulating evidence that eukaryotic gene regulation systems are really operating in a far from equilibrium regime. I would stress that this universality implies that it's not just that a model cannot explain the data, but no model can explain the data if it's at thermodynamic equilibrium. That's the universality. And you can make that conclusion without fitting any of the data. So that's unusual, I think, to be in a position where one can make a statement that's quite as, so these Markovian models, any cellular input-output function, provided the system is at equilibrium, cannot give that data. Okay, so what happens if you're not at thermodynamic equilibrium? And what happens is that the situation is completely different. Sorry. Which way am I going here? Yeah. So this is a very simple system that we're looking at. It's a family of models where you imagine you have a protein which has 2 conformations and 1 binding site. So if you think of the underlying kind of graph, it's a kind of ladder. Okay, so the more conformations you have, the more it extends in the horizontal plane, but there's always only 1 binding site. So you have a kind of family of very simple models here. And if you impose, if you allow the rates in the system to be away from thermodynamic equilibrium and you start plotting position steepness, what you find are the position steepness regions expand to fill up the whole of the positive quadrant. So you can get unlimited ultrasensitivity, sharpness, from a single binding site away from thermodynamic equilibrium. Now, this, I have to say, is really puzzling to most people because the way ultrasensitivity or sharpness is typically interpreted is from an equilibrium perspective, where you think of it as arriving from cooperativities between the molecules, but there's only one binding site. Somehow it is being cooperative with itself. That's really weird to think about. But nevertheless, you can generate unlimited sharpness in this way. So this is really showing that this Hopfield barrier demarcates the equilibrium regime where I think we have a very good understanding and intuition from what's happening, from a regime where actually most of our intuitions start to break down because we end up with phenomena like this. Okay. All right, so I've sort of zoomed through these things here, but I think what's really interesting is the challenges that we have in front of us to start to look at. One of the things we're very interested in is the question of these cellular input-output functions have been steady state, that we assume quasi-static behavior, that these are steady state. But now we have access to more and more data that is time resolved, and we can think about systems which are not in steady state, but are maybe in the process of reaching a steady state. Are there Hopfield barriers in the transient regime? Meaning, if I have a system which is processing information in a transient regime but relaxing to thermodynamic equilibrium, is it fundamentally different from a process which is relaxing to a steady state which is away from equilibrium? And preliminary calculations suggest that it might be. And I didn't talk about this, but when you move into the transient regime, we always thought that this is going to take us away from this nice kind of rational setting where all of the functions are sort of rational functions, because when you're in the transient regime, it looks like you can't use these kinds of algebraic methods to access things, and that turns out to be wrong. You can. And indeed, the rationality reemerges in the transient regime. So that's one set of questions. But I think perhaps the thing that's most interesting is what happens away from thermodynamic equilibrium. And I think this opens up a whole set of things where we have very little understanding of the kind of language and concepts we need to use and in order to provide biology with, in order to reason about information processing away from equilibrium. And I think that's something which I think will resonate with many people here. Okay, so I need to say thank you to my colleague, Angela DePace. And it was really conversations with Angela over a long period of time that started us thinking about this question of energy expenditure in gene regulation, particularly. And the work that I've described has been in part a collaboration with many of the people that have been shared between her group and mine. And Ji-Hye Park, who's shown there, it was her data that I briefly showed you a few slides ago. So that's my lab, and a lot of the work that we've done on the linear framework has actually come from undergraduate research projects, and I've listed some of the people who have made particular contributions to that, but thanks for your attention.